Good afternoon, and welcome to the Brain and Behavior Research Foundation's Meet the Scientist monthly webinar series. I'm Dr. Jeff Brownstein, BBRF's President and CEO. Today, Dr. Catherine Brownstein will present Investigating the Genetic and Biological Mechanisms that Predispose to Early-Onset Psychotic Illnesses. BBRF is the world's largest private funder of mental health research grants, supporting transformative discoveries in order to develop improved treatments, cures, and methods of prevention. The high quality of the research we fund is made possible by the BBRF Scientific Council. This group of 195 prominent mental health researchers reviews each grant application and selects the most promising ideas with the greatest potential to lead to breakthroughs. The Scientific Council guides the Foundation to fund creative and impactful basic translational and clinical research relevant to the whole spectrum of mental health. One reason the research funded by BBRF has such great impact is because we do not limit to one illness or condition. Since 1987, the Foundation has awarded $461 million to fund more than 6,500 research grants around the world and across a broad spectrum of brain conditions, including addiction, ADHD, anxiety, autism, bipolar disorder, borderline personality disorder, depression, eating disorders, OCD, post-traumatic stress, schizophrenia, as well as suicide prevention. Today, I'm delighted to introduce Dr. Catherine Brownstein. Dr. Brownstein is an assistant professor in pediatrics at Harvard Medical School and a research associate in the Division of Genetics and Genomics at Boston Children's Hospital. She was a 2019 Young Investigator. Our webinar will begin with Dr. Brownstein's presentation, which will then be followed by Q&A. To submit your questions, please use the Questions tab on the control panel on your screen. Please feel free to submit your questions at any time. Following the presentation, I'll ask as many as possible in the time allotted. And now, I'm pleased to introduce Dr. Brownstein. Catherine, the floor is yours.
Thank you so much. And you can see my screen okay?
Yes.
Great. So I don't have any conflicts to disclose. And I wanted to emphasize that I would like people to walk away today with just a few things. One is to understand the signs and symptoms of psychosis in youth, and to become a little more familiar with the current genetic findings of early onset psychosis due to research, and review the impact of early life adversity on early onset psychosis, and then understand why studying early onset psychosis is important. So I'm going to start with a brief introduction of Boston Children's Hospital, where I've worked for the last 13 years. It's a tertiary hospital in Boston, Massachusetts, and patients and families come to BCH with two questions: what's wrong with my child, and what's next for my family? And in a lot of situations, genetic testing and clinical expertise can be used to answer this, but sometimes it can't. And when patients and families are on a diagnostic odyssey, a lot of times they want to be involved in research. And my colleagues, Alan Beggs and Dr. Pankaj Agarwal, were noticing that there were a lot of patients interested in research and babies being born in the NICU that had seemingly genetic disorders that doctors and researchers couldn't figure out, and they were ideal for research, but they didn't have a protocol where they could be enrolled. So either it was up to the clinician to write their own protocol, which takes a lot of time and effort, or the patients were being crammed into a protocol that wasn't necessarily the best for them. So Pankaj and Alan designed a new protocol that could enroll any rare disease. And it was philanthropically funded by the Manton Center, the Manton family. And they made a virtual center that anyone with a rare or orphan disease could enroll. And patients could self-enroll, and researchers and clinicians, if they were stumped, they could refer patients to the Manton Center. So the process is they get referred, patients and families enroll, which requires speaking with a genetic counselor to make sure they understand the risks. They collect the biospecimens. A lot of times it's blood or saliva, and can be sent from all over the world. We usually do sequencing. A lot of times now they've already had sequencing, so we get the sequencing sent to us. We reanalyze. We have a weekly meeting, and we're not selfish. If we can't figure things out, we send it to who we think can figure things out. Then if we have a candidate gene, we do confirmation and functional testing, either getting an animal model or protein model or something else. And then if we're fairly certain, we can CLIA confirm it, which means like get it independently validated in a lab that's been certified, and then can have the result returned to the family. And then we always publish so people can learn from this. And this approach has been really successful. So we've had around 6,700 participants already enroll in the Manton Center, around 3,500 families. There's 23 departments of the hospital that have referred to us. All 50 states have sent patients from 53 countries and 6 continents. So if you know anyone in Antarctica, please let me know. So hopefully we could get all the continents. We're referred to by the, and so when I started, I was actually hired to help clinicians get their patients sequenced as a project manager at Boston Children's. And this clinician, the psychiatrist, Dr. Joseph Gonzalez-Hedrick, said that he had a cohort of children with psychosis that he wanted to have sequenced. And I admit that I was skeptical. I had never heard of early onset psychosis. But this first patient, one of the first patients that we enrolled was so striking, I don't think I will ever forget it, the family. So he presented with command hallucinations and behavioral disorders, was described as having mood swings, a huge lack of emotional control and severe separation anxiety, severe self-injurious behaviors, a sudden onset behavioral regression. And he couldn't be left alone with his sister or any animal. And he was found hitting himself in the head and saying that he was trying to get 2 small boys in his head to shut up, and that these boys were saying bad things and trying to get him to hurt himself and others. And he felt that he needed to listen to them. So we did sequencing on the parents and the child, and found a de novo variant in ATP1A3. De novo means that it's not found in either parent, so it's like lightning striking. So you know that usually you get traits from, or one chromosome from your mom, one chromosome from your dad for each chromosome. But here it's neither parent had that variant. But it was in the child. It was like lightning striking. So, and this was in a gene called ATP1A3, and it was a variant that had never been seen before. So the second column is all the databases that have all the genetics that have been deposited there. So if something's been seen before, it would be listed here. There's still a 0. I actually took this screenshot today. And it was predicted as being damaging to the protein. So it was predicted as being something that would impact the protein's function. And the ATP1A3 is wasn't a new gene at that time. It was really interesting because this gene had been tied to various phenotypes, including a form of Parkinson's disease, something called Kapos syndrome, which impacts the eyes, and then alternating hemiplegia in childhood, which really was stiffness or weakness on one side of the body that can affect children, but then it can like spontaneously remiss, sometimes with something like a nap. So this gene had been known to be involved in various neuropsychiatric phenotypes, but presenting as early onset psychosis was kind of new. And then we saw another child get referred to the Manton Center, also with a de novo in ATP1A3 and severe behavioral disturbance. So again, we always want to make sure that what we're looking at is correct and that we can reproduce it. So we were able to get stem cells from a patient and send them to what was then a very talented postdoc, Richard Smith, who's now a very talented professor at Northwestern University. And he was able to first demonstrate that there was a phenotype of the cells in a petri dish, that is, that they looked and acted differently from normal cells. And then he was able to CRISPR correct the variant and show that that corrected the phenotype, and then the cells looked more normal. He was also able to look at the expression patterns of the genes in these cells and show independently that it looked like other gene expression patterns in schizophrenia cases. So we were very convinced that, first of all, that variant was tied to the psychosis phenotype in the child, and also that ATP1A3 is an important gene for brain development. And Richard is still working on this gene. So next we'll go into more about psychosis in youth. So I don't need to tell this group that we all think that mental health is multifactorial, and there are many things that can influence it: genetics, environment, psychological and social causes. And typically you need a combination in order to cause illness. But as with anything, there's a spectrum of phenotypes. And psychosis usually manifests itself in late adolescence and early 20s, but it is possible that you do see it in very, very young children. And there's different types of psychosis. You can have psychotic-like symptoms that are transient, just unusual thoughts or beliefs. Then you have some signs and symptoms of psychosis without hitting the level of being diagnosed with psychosis. Then there's early onset psychosis, which there's different ages that are used as a cutoff. Most, I think, use 19 for early onset and 13 for very early onset. And then childhood onset schizophrenia, which is a stricter definition. Like schizophrenia is different than psychosis. Well, has a slightly different definition than psychosis, but again, we can use the cutoff of age 13 and for early onset, very early onset, and 19 for early onset. So there's a lifetime prevalence of psychosis can range from 2 to 3.5% depending on the study that you're looking at. And then 12.3% of that is before age 18. And then 3% is before age 14. And here again, you see there's a different cutoff for very early onset. Here is 14, sometimes it's 13. But which goes to show that, like, yes, it is seen. And in the Manton Center, with Dr. Gonzalez-Hedrick's patients, we saw patients presenting as early as 4 years of age. There's also various symptoms that can be observed in early onset psychosis. It's not uniform. You can have bizarre behavior, thought disorders, delusions, hallucinations, flat or blunted affect. It's not uniform. And then we started seeing even more cases coming. Boston Children's Hospital is one of the only hospitals around that was equipped to handle these severe cases. So we just kept enrolling. Here, the referral was a 12-year-old boy who presented to neurology at age 5 with concerns about his school performance. He had delays in gross motor skills and some behavioral concerns, but he was above average intelligence. And then at age 7, had concern for ADHD and compulsive behavior and mild social delays. And then age 9, really became evident that he had paranoia and hallucinations, and then was diagnosed with major depression with psychotic features in the context of a nonverbal learning disability and obsessive-compulsive disorder at age 9. Had a main phenotypes were a fear of being hunted, voices listening to him, and a disproportionate fear of mortality. Here we were again able to sequence the patient's parents and the patient, and again was a de novo variant. So again, wasn't in the parents, but was in the child in a gene called TRAP. And I took this screenshot again today, and it still hasn't been seen again or before. It's not in any database and was predicted as being very damaging to the protein. And then since we published it, many other children with variants in TRAP have been published with a range of behavioral phenotypes. And this, we're happy to say, was the CRISPR mouse creation was funded by the Brain and Behavior Research Foundation. And Dr. Smith, who was helping us with the ATP1A3 case, is now taking over that mouse and doing the comprehensive phenotyping. So thank you again. And using this infrastructure, we have created many programs for candidate genes for early onset psychosis, including ZMYM2, ATP1B1, FOXP1, CMIP. And in fact, we had so many cases and were able to create such a cohort that the Fuss family, which had been helping us with the developmental neuropsychiatry clinic led by Dr. Gonzalez-Hedrick, created the EPICenter, the Early Psychosis Investigation Center. And this had the goal of having a well-typed early onset psychosis cohort with biosamples and able to do much bigger studies, much more powerful, be able to compare our cohort to other published cohorts and cohorts of other phenotypes. And this is led by Dr. David Glahn, who came to Boston Children's and Harvard from Yale University to lead the center. So you can see the main takeaway here, I'm not going to walk through every single little piece, but that we have a very huge protocol with lots of different assessments to really get a comprehensive picture of the psychosis in a child, and the phenotypes of the family members. And this enrollment is a little bit out of date. I actually think we're up to 200 probands now, but it goes to show that we have a whole team of individuals that are working on enrolling and doing these assessments, and has been going for a few years now. And we're able to capture some really interesting data here. This is a study of psychotic diagnoses done by Josephine Molin, who's, I think, an assistant professor at Boston Children's now. And showing the breakdown of non-affective probands, affective probands. Affected psychosis means like they coexist with a mood disorder, and their relatives and controls. And really being able to break down and see the depth and breadth of diagnoses and comorbidities that go along with the early onset psychosis phenotype. And as I said before, with this comprehensive and standardized phenotyping, we're now much more able to compare our cohort to other cohorts. And we reached out to David Glahn's longtime collaborator, Dr. Jacques Mont at University of Montreal, who was able to help us do some of these comparisons in his lab. So it's known that there's common genetic variants in the population that influence early onset psychosis risk. That's been published in the last few years, just different elements within the genome that increase risk of early onset psychosis. And then there's also major elements of the genome that confer large risk for schizophrenia. So that are shown to be present at a higher rate in individuals with adult onset schizophrenia than you would expect. So, and these are just some of these risk factors. Some are genes like GRIN2A, NRXN1 deletions, 16p11.2, and then 22q, in addition to others. And so these are genes that are thought to have a large effect, that if you have a variant in there, it increases your risk. But we also know that there's other risk factors out there too. We see a lot of variation in the genome, and the issue is, okay, if you only see an N of 1, is it a coincidence or is it causative? So really, it's a global effort to be able to make these data sets and turn an N of 1 into an N of 5, an N of 10, and really be confident that what you're looking at genetically isn't just a coincidence, just a co-occurring variant in the genome. Like all of us have variation in our genomes, and that it's truly causative. So one thing that we look at in the genome is copy number variants. And this is, again, you usually get 2 copies of a gene, one from your mom, one from your dad. Unless you have an X and a Y chromosome, then you only have, well, 1 copy on your Y from your dad and 1 copy on your X from your mom. But that's not always true. That's a gross simplification. For a lot of genes, you can have a copy number variant where you could have more than 2, or you could have 1, or you can have 0. And depending on where that copy number variant is, it can do absolutely nothing. Like you could be missing a gene completely, like we all have around 20,000 genes. Some might be missing and do absolutely nothing, and you're totally fine walking around without that gene and never know. Or you could have like 7 copies of that gene and it does absolutely nothing and you could be totally fine again. But then there's other genes where if you're missing a copy, it can cause a pretty serious condition. Or if you have an extra copy of that gene or area of the genome, and it could there again cause a pretty serious condition. So here's an example where you have A, B, C, and D areas of a chromosome. And in deletion, you're missing B. In a duplication, you have 2 copies of B. In an inversion, that means the chromosome broke and reattached in like the wrong spot, quote unquote wrong. B and D are backwards. And then you can have an insertion where this unknown piece, E, just came in from somewhere. It could be from another chromosome, or could be just kind of random DNA. So all these things can happen, and you see them quite often, and it's hard to know, like, is this important or is it not? And it's just kind of what makes you you. So when you study the genetics of a condition like early onset psychosis, you're first caught by what is recurrent, the things that happen again and again. And that's not just in psychosis. There are recurrent CNVs that are things that are a little more common. They have just a hot spot in the genome where it just seems like there's something about it that makes it easier to break there and reattach somewhere else. And there are rare variant CNVs, or CNVs, recurrent CNVs, that are seen in psychiatric genetics. So we have a list of those CNVs, and they seem to increase schizophrenia risk. And again, I think I showed this on the earlier slide. Some of them are like NRXN1 and 16p11.2 and 22q11.2, but there are many. And you see recurrent CNVs in childhood schizophrenia. So here you see in comparison to adult onset schizophrenia, you see a lot more of those recurrent CNVs in childhood schizophrenia, so around 12%. And in adult onset schizophrenia, depending on the study that you look at, it could be anywhere from like 2 to 6%. So in our study, where we had 137 cases of early onset psychosis, which again is a slightly different definition than early onset schizophrenia, we did see an increase in the recurrent copy number variants, the recurrent CNVs, compared to both autism spectrum disorder and controls, and it was significant. And this was really interesting because, again, not all of the children with early onset psychosis have autism. It's not just simply that we're measuring the autism and then they happen to have a co-occurring psychosis. So of course they would be similarly looking in terms of the genetics. So here we show that we had more than the control, much more significant, and a bit more than autism spectrum disorder, the children. And even though we see a lot of recurrent, we see a heck of a lot more non-recurrent CNVs. And again, it's a lot of times it's too rare. We just see 1 or 2 cases with that CNV. And it's not enough to really hang our hat that we think is completely true or causative to the patient's phenotype. And one example of this that I wanted to talk about is, you know, a lot of the genetic databases have gotten a lot better in terms of being diverse. Like, it's not just Caucasian males anymore. There's a lot of different ethnicities and races in there, but there's still, we're still not sure we have adequate representation. And so when you see a copy number variant, you don't, especially if it's not in a person who's white, you're not sure if it's just ethnicity or a racial specific variation, or if it truly is causative to disease or a phenotype. So with a lot of these CNVs that are non-recurrent and rare, we're kind of stuck in a way, or we were stuck in a way to study them and actually kind of decide if we thought they were pathogenic or how pathogenic we thought think they are. So again, Sebastian Jacquemont at University of Montreal was really clever, and he figured out a way of kind of quantifying the badness, with a lack of a better word, of a CNV. So he added up what's called the constraint score of each gene contained through them in the CNV, which is kind of a measure of how conserved it is and how tolerant that gene is to variation. So again, just like with copy number variations, where you could be missing a gene completely and be totally fine. You could have a big copy number variant with several genes in it, but like it doesn't matter because all those genes in there, you can be missing them and be totally fine. But some other ones where you could be only missing one gene, but it's really constrained, and that CNV, even though it's much smaller, could be much more impactful to health. So here we're able to calculate this score based on the genes contained within a copy number of the variant and figure out how to quantify the risk of having that CNV. So we were able to compare the risk of having the CNVs in the early onset psychosis cohort and compare it to the autism spectrum cohort and control cohorts, and show that for deletions, the copy number variants in the early onset psychosis cohort were quite similar to the autism spectrum disorder cohorts, and significantly different from controls. And that was exciting. It's less true for duplications but very true for deletions. So what next? We had I think I've been working on early onset psychosis for 10 or 11 years at this point. And you saw we had 137 cases in the patients enrolled in the cohort. It's taken a long time to build up a cohort of that size. But David Glahn knew that there were other populations throughout the world that did not have access and were fantastic areas to learn more about early onset psychosis. He had a relationship with investigators in Mexico City and was able to clone the EPICenter study in Mexico City. We call it EPIMEX. And with the goal of basically creating the same architecture there, with the idea of being able to actually understand the genetics in a Mexican population. So we have proposed EPIMEX, which was to recruit 5,000 individuals from a single childhood psychiatric hospital in Mexico City, looking for inherited de novo variants. And the current enrollment is 729 probands. And again, this is out of date. I think we actually have a few more than that. Controls, close to 800 controls and family members for a total of, well, I know now we're well over 2,000 individuals. And this is the hospital where we have been partnering with to do EPIMEX. We're also fortunate enough that we are able to enroll cases that are children with psychotic disorders and psychotic spectrum disorders. And then we have unique controls in that there are children who are enrolled in the study but do not have psychosis. So they are hospitalized but are at the hospital but do not have a psychotic disorder, which is a wonderful control for understanding the unique elements of the genome that confer to psychosis versus other types of psychiatric disorders. And then we are also able to enroll from the school systems in Mexico City, allowing us for ethnically matched neurotypical control children, which is fantastic because Mexico is not uniform, and I'll show a slide on that later, that being able to make sure that we're matching and using the appropriate control population is actually quite paramount to discovery here. And also by having the leadership in Mexico City, we're able to ensure culturally sensitive diagnosis. And here's an example of how fast the field of genomics is moving. When we wrote this study, we proposed doing a low-pass genome, that is like a very low amount of sequencing across the entire genome, and then focusing on the exome, which is 1% of the genome, but it has the genes in it. So if you're going to do sequencing, it's kind of more bang for your buck to look at the exome. But the price of genomes dropped so much in between of applying for this study and actually being awarded it, that we were able to do whole genome for everyone, as opposed to just an exome and a low pass genome. But, and this is great because, as I said right before now, the ancestry is quite diverse in Mexico. You have individuals with elements of Native American, European, African, Middle Eastern, South Asian, all mixed together. And you want to be sure that anything that you discover isn't just a Mexican subgroup and is actually causative to a condition. And that's really hard when you have such a diverse community. So we're working with Galatea Bio on understanding and making sure that we are matching to control populations in the best possible way. And are accurately distinguishing between what is a variant of interest and tied to the phenotype and what is normal variation. And in this first 806 that we've done, we have a lot of findings already. So actually the first family that I opened up their data, this was staring me in the face, an XP22.31 deletion. And this is, again, one of those recurrent CNVs that you see. And it was, but it's here in a mother and a sister, but because they have an extra copy of the X, they are okay, versus the case who is the patient who is male, and he doesn't have any of the genes contained in that because he only has one X and he's missing those genes. So it's called XP22.3 mycodeletion syndrome. And again, it's one of those known disorders. The second, another one had a paternally inherited NRXN1 loss. We also see it in an unrelated case. And again, I've said this gene name a couple of times already in this webinar as a known gene that's involved in neuropsychiatric disease. Then we're seeing some really interesting de novo variants in newly discovered syndromes. So the date on this paper is in 2021, where they just discovered that QRH1 variants can cause a spectrum, a phenotypic spectrum, including social behavior, speech delay, moderate motor delay, learning difficulties. So this was really exciting to see. Here is called a VUS, that stands for variant of uncertain significance, and they'll say variant of unknown significance, just because it hasn't been seen before. And then you can take that information and look at the potential impact, the type of change in the genome, and again, do functional work, compare it to other cases that were deemed to be pathogenic to make an assessment. And then here is a paternally inherited PRODH loss. And this is a known gene for risk and protection of schizophrenia, though it is a little bit controversial where there's some publications saying that it's not actually a gene that's important in schizophrenia. So it's definitely something we're going to keep our eye on. And then we have other genes that are coming up in that are less well known, but still have a body of literature behind them. Like here, it's a neurodevelopmental syndrome with cerebral and eye anomalies that was seen where we unfortunately didn't have a total trio here. So it's either paternally inherited or de novo because we only have the patient and the mother enrolled. Then we're seeing compound heterozygous variants. Here is an SPG7 where there is a study saying that compound heterozygote variants in this gene can be linked to psychosis. So we are very interested to see this case. And we're also seeing things in the control population. So this is a control from that's considered to be unaffected, but as you see, she has triple X syndrome, where normally you get, again, one copy from your mom, one copy from your dad, and then you get two copies from one parent accidentally, and it doesn't. Most of the time, it's actually, I think 90% of people never know that they have an extra X, but it will be interesting to quantify these types of changes in Mexican population, general and in neuropsychiatric hospital. So we have a lot to do left on this. We're going to continue with our Mendelian disorders. That's, again, looking for genes of large effect or previously unknown. Then we're going to continue also with our cohort analyses, look at the psychiatric diagnoses and controls, non-psychiatric patient controls, then look at comorbid conditions, suicidal ideation. We want to do imaging studies, transcriptomes, that's looking at the gene expression, and then proteomics. We also have studies from our EPICenter cohort, that's again the cohort that we formed at BCH, that we really look forward to looking to see if our preliminary findings hold in our much larger cohort of EPIMEX. So for example, we looked at the average constraint score, so like the badness score of variants in genes that are in the patient but no longer not found in either parent. So again, the de novo variants, and showed that in our early onset psychosis cohort, they were more constrained genes than they were in controls, but it seemed to be driven by the de novos on the X chromosome, that having a de novo on an X really seemed to be in worse genes, so or more constrained genes. So we want to know if that holds. We also compared it to a mixed neurological phenotype. So these were cases where they had like a neuropsychiatric disorder that wasn't psychosis, like autism, and we also included epilepsy in there, where they seem to be much more similar to that than to the control cohorts. So we're really excited to see if this type of finding holds once we have many, many, many more patients than the 137 that we were able to form in our cohort here. So lastly, I'm just going to go quickly through, when you're a geneticist, like everything looks genetic, like hammer and hitting everything, thinks it's a nail. But fortunately, at EPIMEX and EPICenter, we have a very diverse range of specialties that are able to look at all the risk factors of psychosis. And here are some of the effect sizes. This has been published before, that we know have a huge impact. So I want to draw attention to heavy cannabis use. That seems to be quite important, along with adult trauma and parental severe mental illness. And we're looking at here early life adversity and psychopathology in our cohorts to see what phenotypes are associated with higher stress load. Also, the relationship with suicidal ideation and cannabis use. And in EPIMEX, seeing the impact of early life adversity and early onset psychosis and siblings of cases of patients with early onset psychosis. We also are looking at the impact of social disparities, and specifically again on cannabis use and psychosis risk. So in conclusion, symptoms and impairments in early onset psychosis youth are similar to those seen in adult onset psychosis. Cognitive and neuroanatomic deficits are common. CNVs influence liability. Childhood trauma and heavy cannabis use are risk factors. Outcomes are highly variable. Better treatments are needed. We have a huge range of collaborators and colleagues that have been working on this. I want to highlight my collaborators at Boston Children's Hospital and Harvard, especially David Glahn, Alan Beggs, and Joseph Gonzalez-Heydrich, and at Northwestern University, Richard Smith, at University of Montreal, Elise Duard, and Sebastian Jacquemont, and many more. I'm sure as I finish here, I'm going to remember people that I really should have listed on this slide. I want to show this slide to say, if you think that you know someone who would benefit from being enrolling in the EPICenter or would like to enroll in the EPICenter, here is our information. We have an email address and website. QR code here. And then if there's something that's not a rare or orphan condition, it doesn't have to be neuropsychiatric in origin. We do anything that's considered to be rare or orphan condition. We have the Manton Center, and again, patients can self-refer, and here is our contact information for that organization too. And please feel free to email me, and I look forward to any questions. Thank you so much for listening to my talk.
Catherine, thank you for these very complicated scientific issues, and you did a great job explaining it geared towards a lay public. So thank you very, very much. I guess the question that I have in terms of next steps is, in looking at some of these variants, how does that help lead to potential treatments for people? Are you able to look at the proteins that are related to that gene and then see if that's something that maybe could help in terms of a treatment issue?
Yes, that's exactly it. Also, just learning from experience that if one medication seems to work better when on a patient with a certain CNV, then instead of having to go through the trial and error over and over again with the next patient, maybe just try that medication first. Also, the definition of precision medicine doesn't have to be just pharmaceuticals. For example, with ATP1A3 in the children, they found that they really benefit from naps during the day, and they would do much better in school if they were able to have a nap at some point. So it's just learning from experience and learning from the genetics, in addition to, of course, yes, totally, the finding drug targets for new medications and repurposing other ones.
So if a family were to join in the study, obviously there's scientific benefits to the world, but also potentially based on the information that you obtained and other information you already have developed, there may be some benefits to that child in terms of treatment ideas.
Yes, I'm not a clinician, so I am only speaking from what I've heard from the clinicians that treat the patients, but yes, that is my understanding.
Good. And if you were going to say where you see this going, you said you've been doing this for 11, 12 years. What would you tell us in 5 years from now or in 10 years from now about where this has proceeded to?
I really think it's gaining in speed and moving us towards precision medicine and precision treatments and N of 1 therapies, and being able to find the right treatment the first time, as opposed to this odyssey and going back and trying 4 or 5 different medications before figuring out the one that's best. I just think like it's rapidly getting better, just from when I'm started to where we are right now, and it's going to continue at that pace.
Yeah, I agree. I think, as a psychiatrist, back in the day, and probably to a large degree still now, there's no blood test or brain scan to say what medicine to use first, but that's where this type of work will lead us to, or other treatments other than medicine. So I think that is very exciting. I want to ask you a little bit, and you spoke about risk factors, about the interaction between a person's genetic makeup and the environment. Could you talk a little bit about how that works?
That's a fantastic question. So, oh gosh, how do I approach this? So working with early onset psychosis, we're looking at genes of large effect. That means that you are much more likely to have a phenotype if you have a variant in that gene. That isn't a one-to-one relationship. For example, a lot of cases that we see, a parent is actually a carrier of that same CNV, but they're okay. And there you attribute this difference. How can 2 people have the same CNV and one have psychosis and one not? And we attribute it to several things like, okay, the background genetics, the other things that they have going on genetically, and also the environment. We have kind of heartbreaking stories of a parent that was a carrier being prescribed the wrong medication and way too much of a stimulant and having a psychotic break in adulthood. And then the stress of that, having a parent with have a psychotic break caused the carrier child to have a psychotic break. But there again, it was, you want to say, well, maybe it wasn't completely avoidable, but like understanding risk, and that you have to be extra careful with these medications. You have to be extra careful with stress and managing the environment. Again, I keep talking about precision medicine, but that's really kind of where we're leading, that I'm not talking about general environmental genetic risk here. I'm talking about for these early onset cases where they have a known gene of large effect, that minimizing the impact of the environment is sometimes extremely effective. And it goes to show with these carrier parents that have managed fine in life with that same risk factor. and studying that and understanding recommendations for all carriers is one of those areas of research that makes me a true believer that we need to do as much of that as possible to maximize outcomes for everyone.
Yeah,
Sorry, that was.
no, that's a very important topic about sort of the relationship between a person's inherent gene makeup and their environment, and that you could have, the same gene makeup and not have whatever environmental issues, and be okay, but you could have some environmental issue that tips you over into illness. It's a
Yes.
very important issue. And along those lines, I want to ask about the cannabis use.
Yeah.
Could you tell us a little bit more about the information you have from this? Obviously, it's very important as more and more we see more and more legalization of cannabis use around the country. And what are you seeing in terms of that as a risk factor?
Well, I only saw this on the slides as I was preparing for this talk from my colleagues at the EPICenter, and I was shocked actually at how big of a risk factor it was. I, it was right at the top of risk factors, and I feel like this needs to be publicized more, because even I didn't know about it until I was preparing for this, where you know, again, going back to our genetics environmental risk, perhaps it's not, some people really need to be careful. Sorry, I'm giving a non-answer here because it's not actually my area, but my colleagues are definitely seeing its impact in our population.
Well, you gave a good answer, and the fact that you included other information that's not in your particular area of research is extremely useful. I appreciate that. And I think it really does point to some of the risks of cannabis use, for people, in the general population, that if they may be of higher risk, they should be even more concerned about the use of those substances at this point in time. And we know that brains are still developing through the mid-20s. So people should be concerned about that in terms of risk. The last thing I want to ask you is just to say a little bit about these de novo. What is our understanding of what may cause that to occur?
So everybody has, well, pretty much everyone has a de novo variant or a few de novo variants. It's just an error during development where something happens and it's a mistake, and most times development continues on and you're okay. But in these cases, there's a mistake in the genetic code, and that mistake replicates and goes on, and for some reason it can result in psychiatric disease. That's why in research it's so, researchers always try to get both parents and in addition to the affected child, because that's always a great place to start looking when you're looking for something that is causing a phenotype that neither parent has. It's and it's also especially powerful when you have a phenotype as severe as early onset psychosis, where sometimes neither parent has any psychotic tendency or mental health issue. So it really seems to come out of nowhere. And it's just this thing that happened to happen. The other place that we always try to look is recessive, where you need two damaged copies, one from each parent, in order to have that phenotype. And if you just have one copy, so like your mom has one copy and your dad has one copy, and they were both fine, but then you have two copies, and then you are able to have the condition. So just in genetics, just really on a surface level, that's always a really important way to start, where to start looking. And a lot of times that's where we find the answer. But there again, because everyone has a de novo variant, you can't look at something and say, oh, we've never seen this before. This has to be it. It absolutely is it, because everybody has something, and everybody has something and you might have something that's in nobody else in the entire planet. So you have to be sure and have to be convinced beyond a reasonable doubt that that de novo variant is actually causative to disease.
A very good response to a complicated question. Catherine, I want to again thank you for the work that you're doing, the work that you're continuing to do, and for taking the time out for this outstanding presentation. I also want to, yes, no, thank you. I also want to thank everybody who joined us today for the presentation. And I'd ask people to please consider making a donation to BBRF today. 100% of every dollar donated for research is invested in our research grants. And we're able to do this because our operating expenses are covered by separate foundation grants. This means that when you donate a dollar for research, that dollar goes directly to the scientist. To make a gift, please visit bbrfoundation.org or call 1-800-829-8289. This webinar has been recorded. If you missed any portion or would like to share it with someone, please visit the events and webinar page on our website. And please join us again on January 14th at 2 p.m. Eastern Time, when Dr. Christopher Seggi, an assistant professor in the Department of Psychiatry and Brain Sciences at the Medical University of South Carolina, will present Fulfilling the Promise of Non-Invasive Brain Stimulation as a Precision Medicine Tool. Once again, thank you, everybody. And remember, together we can dramatically improve the lives of those living with mental illness and enable more people to live full, happy, and productive lives. Take care.